Hello, and welcome to the FDA public meeting using methods from PFDD Guidance 1 and Guidance 2 as tools for including patient experience data in clinical trials, lessons learned about data collection and analysis. My name is Shannon Cole, and I'm a member of the patient-focused drug development staff in the Center for Drug Evaluation and Research. I will be facilitating the meeting today. This meeting is the second in a series of 2 public meetings. The first public meeting took place on June 30th, and the meeting materials are available online at fda.gov. As we discussed during the first public meeting, patient-focused drug development is a systematic approach to help ensure that patients' experiences, perspectives, needs, and priorities are captured and meaningfully incorporated into drug development and evaluation. As experts in what it is like to live with their condition, patients are uniquely positioned to inform the understanding of the therapeutic context for drug development and evaluation. FDA is developing a series of 4 methodological PFDD guidance documents intended to facilitate the advancement and use of systematic approaches to collect and use robust and meaningful patient and caregiver input that can better inform medical product development and regulatory decision-making. FDA is convening this series of 2 public meetings to share lessons learned and best practices based on use of these first 2 guidance documents, and to hear from stakeholders about how they have used these guidance documents as tools to advance their inclusion of the patient voice in drug development. During the first public meeting that took place on June 30th, we heard from FDA staff on approaches to collect patient input and sampling strategies to identify and gather input from a representative patient population. We also heard from industry stakeholders on their implementation of PFDD Guidances 1 and 2 in their ongoing studies and frameworks. We heard from a patient advocacy organization on sampling and ensuring representativeness in clinical trials. That presentation was followed by a clinical regulatory perspective that discussed FDA's use of patient experience data in review of medical products. The last session of the public meeting was a Q&A session where speakers answered questions from the chat. We didn't have as much time as we had hoped for the Q&A session last time, so we hope to make up for it during today's meeting. During the meeting today, speakers and participants will discuss a range of issues related to data collection and analysis, focusing on lessons learned and on areas identified as particularly challenging for stakeholders. External stakeholders will present examples of how they have used the guidance documents as tools to inform their collection of patient data. Next slide, please. Next slide, please. Thank you. And here is our agenda for today. This meeting will run from 11 a.m. to 1 p.m. Eastern Time. It will be recorded and posted online. This meeting and the first meeting in the series of meetings have an associated public docket in which interested stakeholders are invited to share examples of how they have used the PFDD methodological guidance series to advance the inclusion of the patient voice in the drug development process. The docket is open to the public and will close on September 23rd, 2022. A link to the public docket will be provided later during this meeting. For today, the next item on the agenda is opening remarks, and the rest of the meeting will consist of 3 sessions. The first session will focus on data collection, where we will hear from FDA staff members. The second session will focus on ideas and practice, where we will hear from representatives from industry and patient organizations. Finally, the third session will be a question and answer session where stakeholders are invited to use the Q&A feature in Zoom to ask questions to the presenters. Now I will turn it over to Dr. Teresa Mullen, the Associate Director for Strategic Initiatives, to provide opening remarks. Dr. Mullen, please turn on your camera and begin your remarks.
Okay, I think my camera's on. And so thank you, Shannon. Good morning. I'm Teresa Mullen, the Associate Center Director for Strategic Initiatives in the FDA Center for Drugs. I'd like to welcome you also to this FDA workshop on using methods from PFDD Guidance 1 and 2 as tools for including patient experience data in clinical trials. We are very happy that you are able to join us for this second workshop. We held the first workshop on June 30th, and that meeting was focused on who to ask and how to ask when you're planning collection of patient experience data. And today's workshop is focused on lessons learned about data collection and analysis. And our speakers are going to be talking about a range of issues, with a particular focus on areas that have been identified as challenging. So for example, use of social media in the data collection, managing barriers to self-reporting, and other experiences from the front lines of drug development. The two guidances that we are focusing on are this workshop are part of a series that FDA is developing under the 21st Century Cures Act of 2016. Congress not only directed us to develop these guidances, but also provided us with a definition for patient experience data. This includes data that are collected by any persons, including patients, family members, caregivers of patients, patient advocacy organizations, disease research foundations, researchers, and drug manufacturers, and are intended to provide information about patients' experiences with the disease or condition, including the impact, physical and psychosocial, of these diseases or conditions or a related therapy or clinical investigation on patients' lives, and patient preferences with respect to treatment of such diseases or conditions. The reliability and validity of the collected patient experience data will determine the extent to which it can be used to inform regulatory decision-making. And this is where that series of PFDD methodological guidances come in. These guidances outline FDA expectations for valid and reliable approaches. And guidance 1 is focused on methods to collect patient experience data that are accurate and representative of the intended patient population. Guidance 2 is focused on approaches to identifying what is most important to patients with respect to their experience as it relates to burden of disease or condition and burden of treatment. Guidance 3, which is now issued in draft, is focused on approaches to selecting, modifying, developing, and validating clinical outcome assessments or COAs to measure outcomes of importance to patients in clinical trials. And Guidance 4, still forthcoming, will focus on methods, standards, and technologies for collecting and analyzing COAs data for regulatory decision-making, including selecting COA-based endpoints and determining clinically meaningful change in that endpoint. Taken together, these 4 guidances describe how stakeholders can collect and submit patient experience data and other relevant information from patients and caregivers for use in product development and regulatory decision-making. You'll note the progression of the topics in those 4 guidances. You should also note that the focus of Guidance 1 and 2 includes foundational qualitative work important to address early in product development, to understand what matters to patients and what you may wish to measure in trials. We sometimes hear from patients at FDA that tell us that after the trial has been conducted, that the endpoints measured in the trial did not reflect what mattered most, or it didn't measure enough of the things that mattered in terms of burden of disease. So it's likely that the most cost-effective approach for a sponsor would be to do this qualitative work well and do it early. If drug developers and others are going to engage patients living with chronic disease and ask them to spend time and provide information about their experiences with disease and their preferences related to treatment, FDA would like that to be done using a high-quality approach and make good use of patients' time and effort. We would like the approaches and the methods and the tools to collect that information to be valid and reliable so we can use that patient experience data to the maximum extent possible. I want to thank our presenters and our panelists for joining us today to share their experiences and learnings related to the collection of patient experience data. We're really looking forward to hearing what you have to share, and we're looking forward to hearing from everyone joining us today in this workshop. And now I'll turn it back to Shannon to proceed with our first session. Thank you.
Thank you, Dr. Mullen, for providing those remarks to start the meeting. Now we will transition to our first session of the meeting, which focuses on data collection. The objective for this session is to provide a focused overview of data collection and analysis with an emphasis on practical implementation. First, we will hear from Dr. Selena Daniels from the Division of Clinical Outcome Assessment. She will discuss leveraging social media to capture the patient experience. Now I will turn it over to Dr. Daniels. Please turn on your camera and begin your presentation.
Thank you, Shannon. And as she mentioned, my name is Selena Daniels. I am a pharmacist and a team leader in the Division of Clinical Outcome Assessment here at FDA. And today I will be talking on how to use or leverage the use of social media to collect patient experience data. Advanced slide. So before jumping in to discuss the use of social media, I want to start by setting up the landscape of what the context that we're discussing is use in, which is medical product development. So the first few slides will be related to this context of interest. In regulatory, our ultimate interest is incorporating the patient perspective and benefit risk decisions. So an important part of regulatory decision-making is the careful assessment of patients' views on benefits and risks. And in order to understand the patient perspective, we need to understand the patient experience. The patient experience in a medical product development context incorporates the patient's journey throughout the course of the disease or condition, and it includes the patient's views, feelings, needs, actions, preferences, and interactions within or outside a clinical trial with respect to their disease and its treatment. We refer to this data as patient experience data. This is data that are collected by any persons or intended to provide information about patients' experiences with a disease or condition. It includes the experiences, perspectives, needs, and priorities of patients related to, but not limited to, number one, the signs and symptoms patients experience and how these affect their day-to-day functioning and quality of life. Two, the course of the disease over time. This includes the changes that patients experience in their symptoms and functioning. Three, the patient's experience with the treatments to their disease. And these are the symptoms and burdens related to the treatment. Four, patients' views on potential disease or treatment outcomes and how they weigh the importance of these different possible outcomes. And finally, five, how patients view the impact of the disease, treatment, and outcomes, and their view of potential trade-offs between disease outcomes and treatment benefits and risks. Overall, the patient's journey should be defined from the patient perspective where possible, informed by input from patient partners, and this can be an individual patient, caregiver, or patient advocacy group that engages other stakeholders to ensure the patient's wants, needs, and preferences are represented in activities related to medical product development and evaluation, as well as informed by clinicians. Advanced slide. So the collection of patient experience data is important. It is important for two reasons. One, patients are experts in their own experience of the disease or condition, and they are the ultimate consumers of medical products. And two, patients' experience data can inform medical product development and enhance regulatory decision-making to address patients' needs. Advanced slide. For regulatory purposes, patient experience data can be used to inform the design of the clinical trial, endpoint development and selection for a clinical trial, as well as regulatory reviews, which includes benefit risk assessments. Advanced slide. The agency has given guidance with respect to capturing patient experience data, and this is found in the FDA Patient-Focused Drug Development Guidances 1 and 2. These guidances assist stakeholders in collecting comprehensive, representative, and meaningful information from patients and caregivers. The first guidance in this series addresses methods to collect patient experience data that are accurate and representative of the intended patient population. The second guidance addresses identifying what is most important to patients with respect to their experience as it relates to the burden of disease or condition and burden of treatment. So in the next few slides, I'll present how the advice in these guidances may be applied to the use of social media to collect patient experience data. Advanced slide. So there are various methods that can be used to collect patient experience data, which we discussed in the first PFDD meeting. In terms of selecting appropriate research method or set of methods, one should carefully consider the research objective. We can't stress this enough. In the first two FDA patient-focused drug development guidances, it focuses on three methods: qualitative research methods, such as interviews and focus groups, quantitative research methods, such as survey instruments, and then mixed methods research, which combines qualitative and quantitative approaches, such as survey instruments with open-ended and fixed response questions, or even interviews combined with the administration of a survey instrument with fixed response questions. And these are just examples. Advanced slide. social media can essentially provide another source of data. Data can be collected qualitatively through passive observation of social media discussions or information. These observations can occur retrospectively or prospectively. Additionally, data can be collected actively by prospectively administering a survey instrument in a social media setting. Best practices for designing and implementing studies using survey instruments are also applicable to the use of social media to conduct a survey. Overall, the methods described on the previous slide are also applicable with social media data. You can use qualitative, quantitative, and/or mixed methods research to collect social media data. From a regulatory perspective, social media does have the potential to provide useful data in medical product development, particularly if it includes information on the patient's perspectives regarding symptoms and impacts of the disease or condition. However, like with all data collection tools, there are strengths and limitations, which are outlined in the first guidance of the patient-focused drug development series. And I'll discuss some of these strengths and limitations in subsequent slides. Advanced slide. At this time, we have limited practical experience with use of social media in regulatory decision-making. We are still learning the regulatory utility of social media in medical product development. And it's likely that assumptions about social media will naturally play out as evidence accumulates. Currently, social media may have value in hypothesis generation and signal detection. So for example, natural language mining. And this could be conducted in the form of targeted social media searches, which may be useful during the preliminary stages of a study to complement literature review findings, inform the development of research tools such as qualitative study discussion guides, or as a supplement to traditional research approaches. So for example, one-on-one interviews, focus groups, or expert opinion. Advanced slide. While social media can provide a wealth of data from easily accessible platforms and result in low burden to users, there are limitations related to sampling and detail on clinical and demographic characteristics. For example, there are some sources where there is no documentation of diagnosis unless it's restricted to those who have diagnostic confirmation. The availability or lack of availability of a diagnosis may impact the study design and whether the research can achieve the research goals. It can also impact the reporting stage if the participants are treated and or reported as if they were confirmed with a disease or condition of interest. These are all issues that should be considered when using social media to collect patient experience data. Other considerations for the use of social media includes, one, choosing an appropriate research design. For example, a mixed method sequential research design approach is an option that can further strengthen the depth of knowledge gained from social media data. Two, carefully selecting the social media source. Different social media communities appeal to different segments of the population, and the degree of the community's users' anonymity may affect what users are willing to discuss. Research using social media data should examine a variety of social media networks and communities to obtain data that can be most generalized to the population of interest to the extent possible. Three, using appropriate methods to collect and analyze data. Data collection methods should address potential limitations. So for example, lacking mechanisms to verify patient characteristics, such as identity or diagnosis, and how these limitations can affect data integrity and interpretation. Four, assessing data quality. Verify the content and sources to avoid false information, for example, bots. Five, protecting privacy. While verified patient community data may be used, in some cases, it may be appropriate to allow users to remain anonymous or post under username. So for example, in blogs and in forums, particularly when topics are of a sensitive nature. Advanced slide. So in summary, there is limited practical experience with use of social media in regulatory decision-making. However, social media may be an approach to collect qualitative and or quantitative data to capture the patient experience. If social media is used as a data collection tool, use scientifically sound methods to collect robust, meaningful, sufficiently representative patient input to inform medical product development and regulatory decision-making. You should also consider research design, social media source, data collection and analysis, data quality, and privacy. And that concludes my presentation. Thank you.
Thank you, Dr. Daniels. Next, we will hear from Dr. Michelle Campbell from the Office of Neuroscience and Dr. Lily Gerard from the Office of Biostatistics, who will discuss data collection and analyses, a regulatory perspective. Dr. Campbell, please turn on your camera and begin your presentation.
Thank you, Shannon, and thank you, Dr. Daniels, for your presentation and great remarks that kind of lead our thinking and kind of remarks that both Dr. Gerard and I will be speaking about for the next 20 minutes. And our perspective is really as reviewer perspectives from either the review division or the Office of Biostatistics. And so when we are developing a treatment, what we're really hoping for, what I think about is we're hoping for, rainbows and unicorns, right? We want the best. We want to be able to add and to really put everything into something, to a program, into a development program that is well thought out, that makes sense, and that will actually be successful in the end. Next slide. If you can hit it again, please, Shannon. Thank you. But in reality, we often run into this. I feel like many of us probably who are watching have done this emotion many times. It's when the hiccups come along of what we think is our well-planned design study and something doesn't work out as we hoped. And so what do? How do we handle this? The PFDD Guidance series intent is to really help lay out what we need to see for evidence to support regulatory decision-making, and also look at the practicalness of where, when we do run into reality and the everyday challenges of implementing a study, where we need to think about how we can optimize our collection of data. So next slide. I want to spend the next few minutes talking about study design. And when we do not have potential, when we're not fully thinking about all aspects of our study design, what are some of the challenges that we often see when an application comes in for review or during the discussions through the IND process in the review division? And some areas I want to spend some time highlighting is that when we have that disconnect trade-off between what patients' expectations of a trial and what the trial can achieve, we're going to talk a little bit about that. Additionally, the risk of missing data for endpoints that are intended for labeling, as well as that lack of sufficient evidence to support labeling for non-primary endpoints. And what does that mean and why we need to think about that in our study design. And of course, our uncertainty in interpretation of results. These are just some examples of areas that, if not fully considered when we're beginning to think about a medical product development program, can impede the interpretation ability and the usability of trial data for regulatory decision-making. Next slide. I want to thank Dr. Daniels for talking about why we need to make sure we're collecting what's important to patients. And that is always the critical piece, right? We want to somehow incorporate our patient voice into our development programs. And we know that one way of that a lot of our external stakeholders are interested in is, can we be able to speak of that patient experience into labeling? And so one area that we can see in the review divisions when we're reviewing marketing applications is its high levels of missing data for endpoints that were intended to be labeling to represent an important aspect of that disease or disorder to a patient. But there was insufficient evidence because of that missing data. Additionally, we have seen where we're not getting the sufficient evidence to support the non-primary data endpoints. So what I'm talking about is perhaps there is strong suggestive secondary endpoints that perhaps, had there been sufficient data, an opportunity to have a discussion for potential labeling could have occurred. But the rigor needed and the evidence needed to support a labeling claim was not present in the data. We want to make sure that we're designing our studies to be well thought out, to include all the possible data needed to support any labeling claims that a sponsor is interested in considering. And really that dialogue of what you're thinking about should happen early in your conversations with your respective therapeutic review division. We also know that a poor study design can lead to missing data, and this can impede our ability to use data for regulatory decision-making. Again, if we have data that is significantly missing that is supposed to support a labeling claim, and we're uncertain about the interpretation of that data, it can impede that ability for us to use that data. So when designing your trials, we need to make sure that you're thinking of the rigor needed to evaluate all potential labeling claims. At the same time, we need to balance the patient burden that may happen if we try to want to capture a multitude of information. And so we need to make sure that our study designs make sense, that they're practical, and they'll meet and achieve the goals as outlined in the protocol and with the discussions with the therapeutic review division. We also want to make sure that we are minimizing missing data opportunities. So making sure that you have a missing data plan in place to understand why you have missing data or how missing data will be handled. Opportunities to explore what that could look like, if that can happen in early developments is very important and can be extremely informative when going into phase 3 trials. Next slide. We also want to make sure we are balancing our patient expectations. I think Dr. Daniels said it great when she mentioned how we're really trying to collect data from our patients from their own experience. And that is really the hallmark of all the work that we do, is making sure that we are reflective of their lived experience. But we need to make sure that we have the correct balance in place. And when I talk about this balance, I like to think of it as this balance between the mechanism of action of a medical product, what is important to patients, and what aspect of the disease will change from treatments. This is often critical, and we need to find the right balance. We understand there are many aspects that are important to patients in their everyday life from their lived experience, but we need to make sure that we're measuring the correct aspects of that patient experience and that it makes sense to the medical product program that is under development. So we really want to make sure that balance is happening. When this balance does not occur, this is when incorrect data could be collected and then is unable to help inform a regulatory decision because we've missed the opportunity to collect the correct context of use or the correct aspect of what we hope would be changing from the course of an intervention or treatment. Next slide. The interpretation of data. It goes back to measuring the right thing, as the previous slide is. When we do not measure the right concepts, it can interfere in how we can make inferences on our benefit risk, and is often then not generalizable to our target population. So utilizing the PFDD guidance series and using good measurement to minimize this variability will be able to help make treatment effect more clear and help us in the review divisions as we're interpreting all of the data, really to understand if there is a treatment effect. This is really important to really think about making sure we have good data collection with clear parameters and understanding of the protocol design, particularly when we have a moderate effect that we need to interpret, which of course leads me to our next slide when we talk about, next slide please, clinical meaningfulness. Always an important conversation, the one that I feel we cannot not forget to discuss briefly. So we can't have interpretation of data. If we have that, we also need to have clinical meaningfulness. There's multiple opportunities to assess this during medical product development, and multiple methods should be used. The choice of methodology of how you are assessing clinical meaningfulness can impact our ability, again, to make good regulatory decisions. Or I should say that, if we don't have good methods that are being used, it can just, again, make the more data murky, make us not understand what is being presented to us, and really have to pull looking at all the data. So think about multiple ways to assess clinical meaningfulness. in your trials. And I want to highlight one final opportunity that is highlighted in Guidance 2 in the appendix. Next slide is exit interviews. So exit interviews is highlighted in the appendix. And exit interviews can be extremely useful if it's well designed to answer a specific question. One example in rare disease would be actually clinical meaningfulness. If an exit interview is not optimally designed, again, you hear me talk about when we're having a poor design or lack of optimal design, it can impact informing regulatory decision-making. Additionally, an exit interview should be thought about early, as in all of our decisions in our trial design. When an exit interview is added later into an active trial, this information may be less informative and not as optimal in helping to answer that specific question. We do encourage those who are considering it to submit their protocol and their draft interview guide for review to make sure that we're capturing information that will help inform us make a regulatory decision. Next slide. And I just want to end with future considerations. So as always, you hear us talk about come early and talk to us, and we really mean that. Do come and talk to us so we can have these conversations early in trial design. We can think through together about where there may be challenges and how those challenges can be overcome. And that we encourage, and we would hope by using the PFDD Guidance series, that we can have scientifically sound analytic plans, protocols that address well-developed methodologies, quality and completeness will be able to be addressed because of these well-thought-out plans. I want to now turn over to my colleague, Dr. Lily Gerard, to provide a statistical reviewer perspective with data collection. Lily?
Thank you, Dr. Campbell, and good morning, everyone. I am Dr. Lily Gerard. I'm the team leader of patient-focused statistical support in the CEDAR Office of Biostatistics. I'm going to discuss further from a statistician's perspective. So on this slide, I present a much simplified version of a statistician's wish. Ideally, it would be wonderful to have high-quality measures of patients' health that can be used to support the construction of well-justified endpoint that reflects an aspect of the patient's health that is meaningful. Then valid data can be generated to support statistical analysis, which in the end gives us the ability to interpret treatment effect within the context of use. Next slide, please. So, however, as a statistician, I often find myself struggling with challenges with not having high quality measures, which make it difficult to move forward in the process of analysis and interpretation. Next slide. When submissions come in, we as reviewers often get many hypothetical questions. So I list two common generic examples on this slide. The first one, assuming that content validity of COA ABC has been established for this patient population, does the agency agree that the proposed quantitative validation plan will be sufficient to support the use of COA ABC in the pivotal trial? Does the agency agree that if construct validity is addressed with the COA XYZ, then the instrument can support the determination of efficacy? As you can see, these questions are hard to answer. And most times, statisticians can only provide high-level comments from a methodological perspective that are not specific to the development program. Quantitative statistical evaluation cannot rectify issues or concerns regarding how well a measure reflects all important aspects of a concept of interest. Next slide. So what exactly constitutes a high-quality measure? First, let's define a measure. According to the Patient-Focused Drug Development Guidance 3, which I hope some of you have had a chance to read, a measure is a means to capture data, for example, a questionnaire, plus all the information and documentation that supports its use. Generally, that includes clearly defined methods and instructions for administration or responding, a standard format for data collection,
well-documented methods for scoring, analysis, and interpretation of results in the target patient population. High quality in plain language basically means that we need to do well or to the best of our ability on all attributes of a measure. In the following slides, I will discuss some topic areas that require careful consideration. Next slide. First, I would like to re-emphasize something important covered in workshop 1, representativeness of patient experience data. Regardless of research methods, patients in the study sample should be representative of the target population so that study findings can be reliably extended to the target population of interest. Here's one example problem that we've run into, where the initial patient experience data was generated in patient sample with minimum symptom severity. However, the pivotal study would like to target more symptomatic patient population. Therefore, it's important to establish representativeness before embarking on further quantitative analysis. Next slide. Now, survey research methods are commonly used to collect quantitative data from patients and relevant stakeholders. Other speakers have spoken enough on this point. So to help improve the quality and interpretability of the data collected, it's important to carefully consider the type of survey response options. Here are 3 common types of response options that we often work with. First, a dichotomous or 2-choice response option, such as yes or no, or true or false. Unless the concept is clear, using dichotomous response option may force respondents to choose between a narrow set of response options, resulting in a response that does not completely capture their experiences or feelings. Then we have numeric rating scale, or NRS. For example, a 0 to 10 NRS commonly used to measure worst pain severity. A NRS provides more response options, but depending on the concept being measured, it may be difficult to pick a score, a single score. For instance, what is the difference between a score of 1 versus 2, or a score of 8 versus 9? Then we have the verbal rating scale, or VRS. For example, a 4-point VRS ranging from none to severe, measuring a symptom severity. In the case of a VRS, the response options should be non-overlapping, and differences among adjacent response categories should reflect true differences in the concept being measured. Next slide. Now, standardization is key to data collection, analysis, and interpretation. This slide shows some example considerations. Depending on the trial design and the type of measure used, one should consider standardized instructions and directions for data collection, standardized training for study personnel, standardized environment for participants to perform a task, standardized devices, for example, provide the same tablet for all participants to report responses, and then also standardized case report form. Next slide. Lastly, I want to highlight missing data. As we all know, missing data is inevitable. We should always have a plan to handle missing data, and as statisticians, we always review these plans to make sure that the methods for handling missing data are appropriate. However, I want to point out that it is important to plan ahead and have procedures in place to prevent missing data. To do this, we should gain an understanding on the reasons for missingness. For example, patients do not all experience the symptoms or functional impairment. Inadequate instructions provided to the patients, causing them to have insufficient understanding of instructions and or tasks needed for data collection. Patients not informed about the importance of data collection and how data will be used. The instrument design and/or the trial design increases the patient burden, programming errors in electronic data collection, and also sometimes the reason for missingness may constitute informative missing instead of missing at random. So with that, I conclude my presentation. Thank you very much.
Thank you, Dr. Campbell and Dr. Gerard. Next, we will hear from Dr. Naomi Noble from the Division of Clinical Outcome Assessment, who will discuss inclusive research, managing barriers to self-report. Dr. Noble, please turn on your camera and begin your presentation.
So hello, my name is Naomi Noble. I'm a pediatric psychologist and reviewer in the Division of Clinical Outcome Assessment. And I'm here to discuss inclusive research and managing barriers to self-report. So inclusive patient-focused drug development should engage patients in a patient-centered way. For example, by conducting research in the patient's primary language. Accommodations and modifications to common research procedures may be necessary. Patients, whether they are children or adults, are the primary stakeholders in medical product development. And for this reason, a representative population of patients can and should be able to successfully participate in research that directly impacts them and potentially their survival. Next slide, please. Accommodations and modifications can be made to typical research methods so that all people of all abilities and backgrounds can successfully participate to their fullest ability and contribute their perspective on measuring what matters. There are two broad types of barriers to self-report. The first are research methods which are inaccessible to patients. And I'll speak more in the later slides about ways to modify research procedures to be more inclusive of patients. The second form of a barrier to self-report is when a patient really is unable to report for themselves. An example might be related to child development. We certainly don't expect neonates to self-report. Also, there may be impacts of a medical condition that may make it impossible for a patient to self-report. But patient-focused research needs to be inclusive and patient-centered so that patients can successfully participate to their fullest ability. And so there are approaches that can make the difference between inclusion and exclusion that I'd like to take a look at with you. Next slide. Beginning with language and culture, consider that studies to collect patient perspectives conducted in only one language, for example, English, excludes diverse patient perspectives from non-English speakers. So to begin with, questions should be culturally sensitive in the patient's language or dialect. In addition, it's important to understand how cultural differences may impact patient responses, and exploring cultural differences can be done through data analysis, either qualitative or quantitative. For all studies, conducting translatability assessments early in the study development can help set research up for success. For example, poorly translated surveys can result in uninterpretable data and can also prevent comparisons to evidence that are collected in the survey's source language. Also, it's important to consider that retaining the question format and response options across translations facilitates version comparability. Next slide. When we consider accommodating people with diverse abilities, ensuring the usability of study materials by patients is critical. So for example, for patients with low vision, using written materials with screen readers or in a large adjustable font can help facilitate patient engagement. Another example, for example, patients who experience fatigue related to their disease or condition, it's possible to abbreviate the study to minimize patient participation burden. A possible method to minimize participation burden if you have a reasonably large patient sample size is to systematically divide sections of maybe an interview, for example, among patients for brevity. So these accommodations aren't only supportive for patients who may benefit from modifications, they can help make study participation less burdensome for all patients. But pilot testing study materials with patients for usability can be critical to ensure usability. Next slide. Including children in research is essential. Patients can be children and still remain primary stakeholders in medical product development. So engaging young children who are capable of self-report can be done in ways that are developmentally familiar. For example, using drawing activities or facilitating an interview with props or illustrations. And in addition, of course, for patients who may have limited attention spans or may have difficulty sitting still, abbreviating activities can help include children, but also ensure high quality data. It can be important to plan for parent or caregiver presence or assistance to support a child's participation in research. And it's worth noting here that, well, caregivers generally should not be present during a patient interview. For the comfort of the child and the family, sometimes it's essential, but caregivers could sit behind the child to minimize influencing the interview, either verbally or non-verbally. In addition, research protocols should include a clear plan for the presence or assistance of caregivers in pediatric data collection. Next slide. So when a diagnosis confers developmental and or intellectual impairments, patients who share that diagnosis may have a range of abilities. Some may be severely impaired and unable to report for themselves, while others may be very capable of self-report with or without research modifications for inclusive participation. So when conducting research with a patient community, a diagnosis alone should not be a reason to exclude a patient from sharing their perspective when they are capable of self-report. We recommend that you include patients who are capable of self-report in research that may impact their lives. Many of the research method accommodations discussed in this presentation so far may be helpful for individuals with developmental and or intellectual differences. But inclusive research supports the dignity and self-determination of all patients, and perspectives of patients with developmental and or intellectual disabilities matter in medical product development. Next slide. Sometimes patients really cannot report for themselves, and in those instances, eliciting caregiver perspectives on observable aspects of the patient's health are critical. Those observable aspects may include signs, events, behaviors that were observed by the caregiver. It's important here to note that we recommend avoiding proxy reporting, which is when a caregiver reports on behalf of the patient as if they were the patient. A proxy example might be, how severe was your child's itching today? But an observer reported example may be, based on what you observed, saw, or what was told to you, how often did your child show the presence of itch, such as rubbing or scratching, from the time your child woke up today until now? So integrating caregiver perspectives can be essential to conducting inclusive research. Next slide. Inclusive research practices can also extend to implementing inclusive strategies for public outreach and education to foster patient engagement. And this may help engage patients who are typically underrepresented in research. So for example, if patients have limited internet access, meeting in person at locations that are more accessible for patients may help integrate and include patients. Patients may benefit from personal interactions with research teams to learn more about participating in a study. And so for these reasons, holding educational events and consenting patients to research in locations that are more accessible, again, can help make research participation more inclusive. Accessing technology can be essential to engage underrepresented patient groups, and providing access to study required technology for patients who are otherwise without access can be essential. Next slide, please. So in summary, barriers to self-report may be due to the research methods used and are not necessarily due to a patient's actual ability to reliably self-report. Patient-focused research needs to be inclusive and patient-centered, factoring in language and culture considerations, accommodating abilities with accessibility modifications, and conducting inclusive research with children and integrating caregiver perspectives. Patient-focused research depends on accessible, inclusive practices so that all patient voices can be heard. So thank you very much.
Thank you, Dr. Noble. Next, we will move on to the second session of the meeting, where we will hear from external stakeholders on these ideas put into practice. First, we will hear from Robin Carson from AbbVie. She will discuss development of research study materials, lessons learned. Robin, please turn on your camera and begin your presentation.
Thank you so much, Shannon. And I appreciate the opportunity and invitation to today's meeting and really the opportunity to share some lessons learned. I'm Robin Carson. I'm the Vice President and Head of Patient-Centered Outcomes Research at AbbVie. And I lead a team of researchers responsible for patient experience data generation. So I hope to share some lessons learned from our frontline experience. Next slide, please. The views expressed within this presentation are mine alone and do not represent those of AbbVie. Next slide, please. I wanted to start with a slide grounding in my perspective on the guidance series and just putting the spin in terms of how we put these into practice in terms of PED generation. I think of these guidance series as setting forth very good examples and best practices for the development of rigorous patient experience data and how to translate this understanding of the lived patient experience into patient relevant endpoints. And so much like the other presenters today who described these as building blocks and foundational, they really are linked. And so when we think about utilizing these as good research principles, we start first and foremost with understanding who is our target patient population. And so the guidance one sets forth good principles on that respect, but we stay aligned with our clinical development colleagues and cross-functional teams to truly ensure we understand that target patient population for the development program so that we can align our parallel research on the patient experience to that target group. We then move into understanding what concepts matter most. So a deep understanding of the patient's lived experience. With that understanding and then marrying that with the MOA and the goals of our development program, we identify a subset of concepts for measurement and we think about what is that right assessment. So guidance 3 will obviously focus on how to determine that right assessment, whether it's selection, modification, or development. And then the real importance of implementation and pulling through that patient voice into the development program is where we start to think about that right endpoint definition. And is the observed treatment effect that we observe on those scores meaningful at the individual patient level? So today I'm going to really focus in on guidance 2, but I think it's really important to make sure that we're building iteratively on these principles as we take the patient experience data forth in drug development. Next slide. So when we think about conducting research with the target patient population to identify concepts that matter, I think it's important to consider these 3 main considerations. Now, the great news about guidance 1 and 2, the foundational in the set and those that are final today, is that I think they really substantiate that qualitative research is robust and formal, and it requires these strategic objectives, rigorous study design, and appropriate planning to be successful. So if I think about what's changed with the new guidance series, it's really lent credibility to the work that many of us have done in patient-centered outcomes research for many years. But these are the key considerations that I wanted to highlight, and then I will speak more in detail about study materials. First and foremost, we want to learn from what's known. So conducting background research, taking the time to look into the literature, understanding input from subject matter experts, both internally in your cross-functional team and external as well, from varied perspectives, inclusive of patients and patient advocacy groups. You can even look at the voice of the patient reports that FDA has put out to see if there's a condition or disease report that meets your needs. All of this will give you an early understanding of the disease condition, unmet needs with current therapeutic options, and help you to really focus in on what are the current research gaps. We want to be mindful of patients' commitment and time to doing this research, and we don't want to be redundant if we can avoid it. So it's also important to take those learnings and develop a preliminary conceptual model of the condition that you will continue to iterate and confirm as you move forward in your journey of the patient understanding. Lastly, we consider the competitive landscape and regulatory precedents. So it's important to understand what other companies have assessed in trials, what's been acceptable for product labeling and promotional usage. And before, are there specific existing guidance documents for specific conditions? And so we want to make sure that at the end of the day, our fundamental product and the new solution that we're offering the market is bringing value beyond that of what's already available and truly meeting an unmet need. All of this background research then moves you into identifying that focused research question or set of questions and making sure you think about the right methodology. Think about not only your question to align with your methodology, but the purpose and the intent of use of that data. So Guidance 2 in particular does an excellent job of outlining sort of the menu of potential methodologies and key considerations for selection of the right one. I think this truly demonstrates that one size does not fit all, and that there's not one preferred method for all intended uses. Again, grounding yourself in the research question and intended use is critical. So for example, if your goal is to think about the concepts of interest to inform selection, modification, and development of a COA that you'd like to put into a future development program, we likely would lean into using one-on-one interviews or focus groups. However, perhaps you want to obtain an early exploratory understanding of what symptoms might be important to patients for hypothesis generation or that early breadth of understanding. Perhaps we consider social media listening. And I think the guidance does a nice job of talking about mixed methods within trials, but I did want to highlight that in practice, we often utilize different methods across several studies, which can help with that holistic understanding. So for example, we might consider early social media listening for that really breadth of understanding, hypothesis generation, early conceptual model to supplement what we learn from the literature, followed by in-depth one-on-one interviews, and then perhaps an observational survey study to replicate those findings in a larger sample and allow for quantification of findings on key research questions. So I tend to think of it as an iterative approach across studies where you refine the questions based on that cumulative understanding. We just heard an excellent presentation about methods to ensure diversity and representativeness. And so I did want to mention that this is critical in developing your study documents and designing your research as well. It's really important in practice that you stay in alignment with the trial and development program, and particularly now as many companies are developing diversity plans to put in place for trial recruitment and retention to ensure equitable access to enrollment in clinical trials. We need to stay in lockstep with that and make sure that we're aligning that sample with the target patient population. Traditionally, we've conducted early phase qualitative research in the US, largely due to infrastructure. FDA sometimes has been largely the first group to review submissions historically. I think we see this changing. In the past, we've leveraged the linguistic validation process to engage with patients globally in those target countries, languages, and dialects to ensure consider for the needs of a particular program, if that's the right approach moving forward. Of course, as you consider global considerations, this can be cost and time prohibitive, and it's always important in practice to balance speed and comprehensiveness. And so one way we've been able to mitigate our understanding if we expect true differences across populations is to leverage global thought leaders. And I'd like to see more and more patient engagement early on in that stage to inform whether there's truly something to be expected in the way the disease presents or cultural perceptions and preferences earlier on to inform that subsequent research. Next slide, please. The guidance talks about the importance of study materials, and so I will briefly touch upon the importance of this. Again, robust planning and the appropriate time set aside to develop these materials is absolutely critical. When creating your protocol, it's really important to think about adverse event reporting procedures, development of screening forms that are aligned with eligibility criteria, and your sampling strategy to ensure that you'll have that robust monitoring plan for your recruitment targets for your inclusive and comprehensive patient population. When doing the interviews, if you're pursuing interviews or focus groups, we really need to think about the mode. We've talked a lot about remote health in the most recent years and the adoption of connections for e-health opportunities and visits. And I think that's an opportunity to really leverage both telephone, of course, interviews, but video as well to allow for understanding of those nonverbal cues and make that connection with the patients. I think there is an open question, though, that we need to think about: has this rapid adoption of remote engagement truly changed our ability to reach a more diverse population? And I hope we'll learn more in the coming years on that front. We personally have not seen a major shift. We've been able to reach all populations of interest, but it will be interesting to continue to evaluate that and make sure that technology is enabling more inclusiveness. We obviously want to consider the interviews or trials of trial participants. So the timing of interviews, are these general population patients or trial participants? When you're developing that protocol, the timing matters. And particularly, we heard earlier about the value of exit interviews. And I do think that when done early enough, these can truly help design a more patient-centric subsequent trials, such as your phase 3. But you do want to make sure that you are developing these in a way that they don't in any way hinder the execution of the core clinical trial and avoid burden on the study staff. I do want to touch upon the interview guide and discussion guide, as this is critical for the engagement with patients. So first and foremost, in our experience, we always want to use trained interviewers. And I have found that it's really beneficial if those interviewers are part of your subsequent project team that are helping you evaluate the data. So qualitative data comes in text, very heavy with quotes, and it can be, volumes and volumes of quotes. But trained interviewers coupled with a good coding dictionary and data analysis plan are trained to look for themes, and with the coding can identify the right categories of interest. And I think it's important not to over rely on frequency counts or just that quantification of it, which we as researchers tend to do, but to have that breadth of understanding that the trained interviewers were able to gain with the engagement with the patients and have illustrative quotes alongside the information. I think it's also important to note in designing your qualitative research to think at this time of how to understand meaningful change and set yourself up for success in what I consider key building blocks. So you may need to debrief those global items. You may want to think about the response options. And of course, if you are conducting something like an exit interview or a survey, talking to patients who've experienced some change in the context of a trial would be beneficial, coupled with your psychometric analyses work. So I think bringing the meaningful change conversation earlier into development will help us truly obtain that mixed method and deeper understanding of what's meaningful. I do want to just talk about the data analysis plan and coding very briefly. I think you need to have it driven by your research question. And certainly one thing that we've thought about is always with the end in mind. Think about how you want to visualize that data. Qualitative research, as I noted, can be volumes of quotes, and we want to categorize it. And it's always important to think about perhaps looking at frequency, but then charting that against perhaps the average bother rating. So having these additional exercises in the context of your interviews will truly help you think about and set up what that core set of concepts that truly matter that you might want to take forward into drug development. Next slide, please. I wanted to touch very briefly on a case example in irritable bowel syndrome with constipation. This is an example where we were seeking to understand the patient experience. And why I share this is I think that it illustrates a lot of really good principles and applications of what's put forth in the guidance in terms of good patient engagement and input. But there's also some key lessons learned. The first and foremost is that this was built on extensive background research that had already been conducted, but we continued to develop and engage directly with the patient population, and we sought saturation, this idea of no new concepts or no new information on the core set of symptoms. So there was really just a robust evidentiary basis. Also, the multi-stakeholder effort. I think everything that we do, whether it's internal at a company or externally, needs to have that cross-functional lens and multiple perspectives to be successful. And then, of course, having the opportunity to engage and align with the FDA as part of the Critical Path Institute Pro Consortium was successful. My next slide is my last, and I'll wrap up quickly. So if I were to reflect on just the key takeaways in terms of our best practices and lessons learned, I think as we think about qualitative research, we need to plan early and allow sufficient time for development of study materials, particularly around the recruitment plan, making sure you have the right patient population, a robust interview guide or research plan, and of course, the coding and data analysis. We also can leverage these guidance series for best practices and guiding principles. We in industry, one of the biggest challenges we face is getting feedback in a timely manner and balancing that with our development program timelines and not having delays in any way of bringing these products to market for patients who need them. And so I think as good principles, the guidance series is a nice way to risk mitigate, coupled with the experience of your team and the research group, to make sure that you're truly proceeding in a way that hopefully can have very predictable outcomes and be useful in the context of regulatory decision making. And then lastly, collaborate with both key internal and external stakeholders to make sure you're optimizing the value of the evidence, and in particular, making sure patients are part of that conversation throughout the program. Thank you so much.
Thank you, Robin. Next, we will hear from Tom Wilgoss from Roche. He will discuss reflections on the utilization of social media data and industry perspective. Tom, please turn on your camera and begin your presentation.
Thank you, and hopefully you can all hear me okay. I have a bit of a cough today, so hopefully I'll get through this without too many interruptions. Firstly, thank you so much for allowing me to talk today. I'm Tom Wilgoss. I work in the patient-centered outcomes research team at Roche, and I've been very passionate about the use of social media data, certainly for the last 10 years or so. And I'm going to talk a little bit today about how we're using social media data in our work, to reflect a bit on the PFDD guidance and how that's influenced the work we do today. And I'm just reflecting on Dr. Daniel's overview at the start of the session. And I think it's fair to say we're all learning. And so this is an attempt to share some of our experiences so that we can continue to learn together. Next slide, please. I also have a conflict of interest to declare. These are my own views and not those of Roche, and I'll let you read the rest of that in your own time. Next slide, please. Just a brief agenda. So I'd like to reflect a little bit on the recent history of social media data in relation to clinical outcome assessments, and highlight some of the key opportunities that we see. I'll also provide a recap on how social media data use is described in PFDD 1 and 2. I'll provide some examples of how we're using social media data in our work at Roche. And finally, I'll wrap up with some reflections and perhaps some unanswered questions for the Q&A or for future discussions. Next slide, please. So, as you're all probably very aware, social media is a rapidly evolving space, both in terms of its reach and also how it's influencing health research. As of this year, there's approximately 4 billion social media users across the globe, and the average person is predicted to have more than 9 different social media networks. So it's no surprise that researchers in our field are excited at the prospect of the depth and the breadth of the insights that could be generated from this resource. However, while the space is evolving and growing rapidly, it's often been concluded that the lack of regulatory guidance on this topic has remained a barrier. I was actually involved in a forum on this topic in 2019, where scientists from across pharma, consulting, and academia reached that very conclusion. So the PFDD guidance in 2020 and 2022 has very much come at the right time. So I did a simple search of PubMed, as researchers sometimes do, to illustrate their hypothesis. And I think it provides a nice illustration of how much growth there is in this space. So you can see over the last 4 years or so, there's been approximately 50 publications a year relating to the use of social media in patient-reported outcomes research. And we're already midway through 2022, and it looks like we're going to reach a similar figure. But there's clearly a trend here in terms of interest in the use of social media data and how it influences our work. Next slide, please. So despite this rapid rise, it definitely remains an untapped space and an unexplored space. And Dr. Daniels highlighted some of the really important limitations earlier, but I also wanted to flag some of the opportunities. One of the things that researchers have found is that social media can provide concept elicitation into anonymous forums that allow people to discuss embarrassing topics, things that they may not be comfortable sharing in a one-to-one interview or in a focus group. I was actually involved in a study in 2017 that compared traditional methods versus social media data and other online approaches. And it was really interesting to see that while there were significant overlap in terms of the concepts that were identified, some of the embarrassing symptoms, or what we would deem to be embarrassing symptoms, were only discussed in social media forums. The other thing to highlight here is that patients are leading the conversation on social media. So those dominant topics can actually tell us a lot about what matters most to them. Social media data also offers lots of opportunities for automation through passive data mining, through smart algorithms, and all of this increases the speed of insights generation. And that's really attractive, I think, to researchers in this field, and even the prospect of real-time data, which is something that hasn't really existed up until the invention of social media when it comes to insights generation. So when you consider the vast amounts of data that are available, combined with the speed that we're able to access data at, it's a very compelling proposition for researchers. And finally, and perhaps more importantly, social media data allows us to collect data from global samples, often in their native language. So this does potentially open the door for more representative and diverse opinions and populations. And again, this might allow for the inclusion of perspectives from groups who may not always be included in the more traditional methods, as Robin sort of alluded to in some of the earlier comments. Next slide, please. I just want to take a moment to reflect on what is actually referenced in the PFDD documents or guidances and why it matters. So as Dr. Daniels outlined, PFDD 1 and 2 were the first documents to really discuss the use of social media data in relation to clinical outcome assessments. In Guidance 1, social media and verified patient communities were cited as potential data collection methods. And here it was called out as a complementary method to some of those more traditional approaches, but also as a potential early insights tool to help shape future methods, such as developing interview guides. Importantly, Guidance 1 also discussed the strengths and limitations of different online methods in a really constructive way, and even offered some potential solutions to some of those barriers. In PFDD 2, which was published in 2022, social media data was again referenced, but in this guidance, there was a specific reference to quantitative methods, something that opens the door to perhaps more wide-scale data mining approaches and prospective online surveys. It also discussed some of the practical considerations for using social media data, including research design, the source of the data, analytical methods, data quality, and data privacy. So they really have been a major step forward in the use of social media in this field. Next slide, please. So perhaps the main focus for my presentation today is to share some examples of how we're using social media data at Roche. And these case studies are certainly not unique, and they probably represent how many researchers in this industry are utilizing social media data. But one thing to flag is that this really is across the whole product lifecycle. So even in the preclinical space, we're seeing social media data being used to help identify unmet need for new indications, but also in defining subpopulations for future research. One area that I'm really passionate about and where we've seen particular utility is in early concept elicitation and in the development of conceptual models. So in both Parkinson's disease and in Angelman syndrome, we've been able to successfully utilize social media data to complement traditional methods such as interviews with patients, families, and caregivers to help identify concepts and outcomes that matter most to patients and families. And one of the things that I think is particularly valuable here is the ability to add layers of quantification on top of the rich qualitative data that we can generate. As all researchers are trying to do, we're often trying to prioritize and identify what matters most. And so having some quantitative data can be really valuable in ensuring that we have confidence that we're targeting the right concepts and the right endpoints. And that really echoes what Dr. Mullins outlined in her opening remarks in relation to getting the endpoints right. We're also using social media data in the selection of clinical outcome assessments, including confirming the relevance of items, but also using it to explore language and terms that are being used by different communities. Again, it can be really complementary in terms of the inclusiveness of the patient voice. It's also enabled us to look across multiple indications to identify concepts of interest that might lend themselves to standardized measurement approaches. Again, this is something that it can be done readily from a desk rather than spending a lot of money and recruiting patients from huge samples. We're able to do some of this early landscaping in a much more efficient way. And finally, the safety scientists have been using social media data for a while now to explore risk characterization for new products in the real world. And the ability to monitor using social media in real time, I think, is something that's particularly attractive to safety scientists. And it's certainly an area where there's been increasing growth in this industry. So these are just a selection of some of the internal case studies that we have. And I think they all represent kind of first steps, first forays into the use of social media in this field. But it's something that is continuing to pick up pace. And from talking to colleagues across the industry, we are seeing greater interest in these methods and something that we're looking to explore further. Next slide, please. So yeah, I guess I wanted to really highlight today that the PFDD Guidance 1 and 2 has been a major step forward in terms of the pragmatic use of social media data. It provides acknowledgement that these data are acceptable, assuming they're robust and we use the right approaches, and complementary in terms of patient experience data. The inclusion of quantitative social media data, I think, is a really important step forward and has given us the ability to generate further insights. Also, the discussion around the strengths and limitations of different methods was very welcome. And I think this is something that as researchers and as people who are trying to make decisions on which methodology we should consider, it's really valuable to hear FDA's perspectives on these strengths and limitations so that we can actively try and overcome them. But of course, there are many open questions, and that goes for all of our research methods, but I think particularly social media data. There are certainly open questions around whether social media data alone would be enough. And I think based on the current guidance, the assumption is that it probably won't be. But as social media data evolves and as our methods evolve and our confidence evolves, is this likely to be a future outcome that we can work towards? I think we're also really interested to better understand how FDA and other regulatory agencies use social media data in their reviews. What are their expectations around methodology, around data quality, around analysis plans, and even in terms of how they expect to receive that data for their review? These are questions I think that would be great to discuss in the future. And then finally, I think it's really important that we highlight data privacy and ethics on the use of social media data, because it remains a complex and ever-evolving space. So as Dr. Daniels shared, we are all learning, but I think this is set to be an increasingly important and influential data source as we move forward. Thank you.
Thank you, Tom. Finally, we will hear from Dr. Kara O'Neill from Cure Sanfilippo Foundation. She will discuss Sanfilippo syndrome study of caregiver treatment priorities and unmet need. She will share her perspective from the experiences of a patient organization. Dr. O'Neill, please turn on your camera and begin your presentation.
Thank you, Shannon. And thank you all for having me here today to share on Sanfilippo syndrome caregiver preference study and how the many principles contained in the PFDD guidance has informed our work. Next slide. Thanks. Sanfilippo syndrome, just as a background, is quite a rare disease. And so with rare diseases comes a lot of challenges that more common diseases may not face. This condition has kind of historically been thought of as a neurocognitive condition, but I think it's important to point out that the accumulation of heparan sulfate, while particularly toxic to the nervous system, also affects every other system in the body. Next slide. And next, I'd like to give you context about why we decided to use our small nonprofit resources, which are certainly precious as we contribute to research, why we decided to use it in this space and why it was important. And that's because we saw clinical trial program closures happening and also hearing from the community that, wow, we thought that that was benefiting us. We saw these improvements, the primary investigators saw these improvements, and many of these were in non-cognitive types of symptoms. Now, typically, cognitive symptoms are the primary trial endpoint for efficacy in these trials. And it really left us wondering whether, these were failed drugs or just endpoints that were not reflective of the patient preference and the ability to make change in this disease within the population that was studied. We also know that most patients are symptomatic at time of diagnosis, and that it was really important to go back and take a look again what was important in this population. And if cognitive outcomes were going to be very difficult to change in the population that was symptomatic, we needed to find relevant outcomes that would allow them to access quality of life improving treatments. Next slide. I shared this timeline with you because when we were developing this study, FDA was in the beginning stages of gathering information about patient-focused drug development and hosting workshops. We partnered with RTI International, some really wonderful social scientists who are experienced in the field and had worked with rare disease populations before to help us with this study. And they had been attending and contributing to these workshops. So while the guidances were not actually out at the initial part of our study, we did benefit from the principles that were being discussed. And we also benefited from a meeting with the FDA where they really provided some very helpful information and things for us to think about in designing the study as we move forward. Next slide. So our approach was to develop a multidisciplinary study team. Again, social scientists from RTI International, some industry patient advocate representatives so that we could understand the challenges and the needs from that perspective, parents, of course, and an expert clinician. And then our external advice from the FDA. We used a method approach, and really the idea was we want to gather as much information in as many different ways as we could so that that would be accessible to gather input from many people in a way that they found comfortable. Next slide. So sampling is something that's talked a lot about in the early guidances, and probability sampling is a particular challenge for rare disease groups who may not have access to all of the patients to really be able to get that nice sampling frame. So we are often left with non-probability sampling, which is convenience. And in our case, additionally, we use snowball recruitment. And while there are limitations there, it's what we can do. And I think we are able to deliver a clinically relevant sample. These are people that are plugged in somewhat and are available and interested in participating in research and clinical trials. Next slide. So to overview who participated in our study, we had a very nice sample size for our rare disease. And I think what is important is that we were able to get perspectives from people across the range of disease from age 1 to 40. And so a lot of different severities, subtypes of the disease that reflected the prevalence distribution, and across the United States and many other countries. We did have limitations with diversity and female caregivers as the reporters, but we still feel that we were happy with the feedback. Next slide. This is an overview of the whole study, which really was broken down into 2 phases. In phase 1, we were focused a lot on understanding that unmet need and prioritization of what symptoms should be addressed by a future treatment. And in phase 2, we took what we learned in phase 1 and wanted to see how that could inform the selection of outcome measures in future clinical trials. And that gets into some further guidances, but this is something that FDA really encouraged us to do, to translate that learnings from phase 1, and we appreciated that. Next slide. So look at phase 1 in more detail. Next slide. In phase 1, we started with in-person focus groups. And as you've heard, they can elicit some very rich and nuanced information about the human experience. Group settings really can allow discussion to build upon itself and reach saturation of important concepts more readily. We used a semi-structured framework for group facilitation and facilitated interview in these focus groups. But I think it's important to touch on some of the things that other speakers have highlighted, which is using an experienced facilitator or interviewer. We're asking parents to think about and consider some very emotionally difficult things about their children and about their lives. And so having someone well equipped to provide a supportive environment is important to really being able to get the full information. We also began and ended these sessions with positive reflection, which gives you another perspective on what's important to families. What they told us was that there are maybe some more narrower aspects of neurodevelopment that are important and that should be focused on, as well as a variety of non-cognitive disease manifestations. Transcripts from this focus group were coded and broken down into these 2 very large domains of cognitive, behavioral, psychological, and physical health impact. Next slide. When we think about meaningful benefit, I just want to point out the challenge in rare disease, particularly a terminal pediatric rare disease, where you have no treatment, and having to consider what's the least amount of change that will be meaningful to you can be difficult, and that the granularity of that was beyond our study. However, I think we can take from this that probably what others outside of the lived experience assume would be meaningful is not exactly the same as what would be meaningful to families. And they want you to understand that very small things can have a very big impact. And one example is sitting and enjoying something for 3 more minutes. So it seems minor, but that's huge to a family, and something they would be willing to risk an invasive medical procedure for. So that really tells you a lot. Next slide. We then went on to the prioritization survey, which included all of the components that you see there. I want to focus more on the best worst scaling activity, because I think that's a neat tool that was really effective for us. And this is also something that was used in the focus groups, and we got their input on it and developed some validity around that. And that helped us understand also if it was going to be feasible to use in this online survey. Next slide. So this is what a best worst scaling item looks like. You cannot impute answers in this type of format. And so within a sample set, you do not have missing data. So I think that that's nice, and that you can kind of have a confidence that you've gotten all the information around these set of attributes. So in this type of item, you take important attributes which are relatively similar, and the computer will then generate a series of these sample sets in which the parent will have to choose what is most and what is least important. And when they are all complete, that gives you relative importance of each attribute. Next slide. And here you see the results of our broader online survey prioritization. Each activity started with some demographics, so we were able to understand the age of the participants, which is important. And also, it was surprising to look at, pain being one of the top important symptoms to treat, and not something that had been addressed in previous trials. Next slide. So takeaways, in-person focus groups are really rich sources of information, but need to be conducted facilitator, and that the best worst scaling construct was well understood and can be a benefit in avoiding missing data. Next slide. Okay, so we'll move on to phase 2. And in phase 2, we wanted to look at these outcome measures. The advisory group identified a set of 34 outcome measures that matched up with the domains of importance. And then through deliberation, narrowed that down to 15 based on their historical use in Sanfilippo, relevance to the disease, and feasibility. Next slide. But we needed to narrow that further. So in phase 2, we used these two activities, asynchronous focus groups and interviews. Next slide. So this is our entree into social media research, and it was a very limited use. We really just wanted to get some feedback on those outcome measures to help us narrow them down, understand which ones might be most relevant to get more detail on in the one-on-one interviews. And of course, others have talked about the benefits and the challenges with social media, and they're the same here. So overall, we felt like this was a good use for getting some pilot data and feedback. Next slide. In our one-on-one interviews, which were conducted through Zoom, we provided the outcome measures to participants ahead of time to look at, or they could review them while during the interview if needed. We asked them to think about the domain and how it presented itself in their child, and if the outcome measure reflected their experience or would be a good fit for assessing change in Sanfilippo. And I'll focus a little bit on the eating and swallowing one. Next slide. And I love the name of the scale, CHOMPS, it's very fitting. But interview participants endorsed that, yes, this domain was very meaningful and relevant. They also felt overall that this was a good measure. But I think what's important is that they can always go back and look and say, okay, this, if this was there, it would be a better fit for our disease population. And they identified things like overfilling of the mouth, pacing of eating, how parents were maybe supporting the child in the eating, and whether they needed more or less support would be important. Also, one universal thing that we tend to see in outcome measures is the lack of acknowledgement of regression. None of these measures seem to be made for degenerative diseases, and understanding where a patient is in the trajectory of their disease can sometimes be really important for interpreting context and expected outcomes. Next slide. So in conclusion, certainly the data demonstrate a multi-system system disease impact and the relative prioritization of treatment targets by families. They endorse that quality of life treatments are incredibly important, irrespective of impact on global cognitive ability, that they have a high risk tolerance for modest benefit, and that caregivers are really very good at identifying adjustments needed to outcome measures and should be use their expertise. Next slide. And very broadly, in rare disease preference work, there are challenges in addition to just the overall challenges, and resource challenges are key as nonprofits are doing a lot of this work. We don't have a lot of big pharma in our space, or none. And so we are limited by that as far as what we can do to reach broader groups of individuals related to cost and resources for translation and many other aspects of reaching individuals. There's also a high disease burden and caregiver impact. And I think this is particularly key in pediatric diseases, to remember that there are all these other factors around caregiving that make it difficult for people to participate and even think about the future of these diseases. And over time, we hope that treatments will emerge, and we know that continued work will need to happen. So thank you for inviting me here today and for the work that FDA is doing to advance patient-focused drug development.
Thank you, Dr. O'Neill. Now we will begin session 3, our question and answer panel, to discuss what was heard during the meeting today. I will turn it over to Robin Bent to facilitate this portion of the meeting. At this time, all panelists should please turn their cameras on.
Great, thanks so much, Shannon, and thanks everyone for attending today, and also for submitting some really great questions. And so, and of course, thanks to the panelists for some really informative and interesting presentations. I think it's been a really great to kind of hear current thinking right now. Before I get started and share some of the questions that we've received and kind of turn to our panelists and ask them for their thoughts, I did just want to remind everybody that kind of in this public meeting forum, that our feedback here is really non-binding, and all questions for specific development programs really should be directed to the appropriate review division. So we'll give you kind of our off the top of our heads thoughts, but please remember that any questions that are specific to a drug development program should be discussed formally with the review division. And so now, just to get us started, I'll start with the first question that we received. And so this was back in the beginning when we were listening, I think, to Dr. Daniels. But how do you use completion rate for evaluating quality? Do you suggest stopping evaluation at a certain point? I saw a company, I can't remember which one, stop analyzing data when missingness reached 10%. And so let me see if I have any panelists that would like to kind of take a thought at this. Tom, I know that you kind of deal with maybe this on a regular basis. Do you have any thoughts that you would be willing to share?
Sure. Yeah. No, I think it's a really good question. I mean, clearly the cutoff rates are arbitrary to some extent, but they matter in terms of being able to evaluate the data. I do think the focus really should be on what may be causing low completion rates, because there's lots that we can do to reduce that. So making sure that the endpoints and the measures are appropriate and meaningful is number one, and that's been called out today. But also making sure that the trial designs are developed with patients and families. We know that completion rates can be influenced by fatigue and just by poor planning on our part. So yeah, I think those are things that are maybe worth focusing on. I can't comment specifically on cutoff rates. I know that we have to sort of preset these for a certain analysis plans and that they do matter. But yeah, it's an ongoing challenge, clearly.
Great, thank you. I don't know, does anybody else have any other thoughts that they want to share regarding this question? I'll open it up to everybody. Don't have the ability to totally see everybody, but I don't see that we have any additional comments. So thank you, Tom. I think that was a really helpful and kind of thoughtful response. Let me move on to our next question, which would be, can you please comment further on how the mechanism of action of the study drug should inform a study about patient experience data? And I think this came in around during Dr. Michelle Campbell's presentation, and so maybe we'll start with Michelle and then open it up to others after.
Yeah, thank you, Robin. So it's important to remember that sponsors should specify and define concepts such as signs, symptoms, and impacts that are relevant and important to patients and that are likely to demonstrate meaningful and interpretable changes in their planned clinical trial. When considering a potential selection of COA support trial endpoints, it's important to have an understanding of what the medical product mechanism of action is. From that, the mechanism of action, what aspect of the patient's lived experience is expected to change from this treatment or the intervention during this trial? How and when could this be assessed? Knowing this information plus what the patient experience is will better inform the trial design. We need to keep in mind that what may be the most important thing to patients may not be what will change due to the treatment or intervention because of the mechanism of action or the trial design. However, the second or third most important concept to a patient may be what is going to change and will be the concepts to be measured during the trial.
Great, thank you. Does anyone else have anything they want to add to that question, or any thoughts that they have related to that, maybe real life experiences or anything?
Robin, I'd be happy to comment on that as well from the industry perspective. So I think what Michelle said is exactly right. And it's really important as we try to also aspects we do not expect to change. Now, early in development, your considerations might be different because you may not know as much about your product. However, if it is known and the mechanism of action doesn't lead to potential changes in certain things, sometimes at a minimum, you may want to capture it if it's a part of the core condition, just to ensure it doesn't worsen. However, I think having a much more focused approach to measurement aligned with what you expect your product to address is actually a better use of your patient's time and really helps balance that burden. And I also think it's really important to evolve that understanding, right? A lot of times companies are developing a product in different conditions, but perhaps related. And so thinking about those learnings across development programs and building on that knowledge and sharing across teams would be really helpful to make those future studies much more patient-centric as well.
Great, thank you. I think that's a really good point. Does anyone have anything they would like to add? All right, so seeing that thing, maybe let me move on to our next question, which came in again, I think during Dr. Gerard's presentation. Are there suggested reasons for missing this to collect?
Hi, this is Lily Gerard. So I'll attempt to answer this question. If I understand the question correctly, I think, first I want to point out that the reasons for missing this listed in my presentation are examples that we have seen, and every trial has its own unique challenges and considerations, right? So I think sponsors can collect reasons for missing this during the qualitative research, or also include as part of the quantitative data collection, possibly a list of possible reasons along with an open-ended response option for other reasons not listed, that together would help identify possible changes to data collection protocol to reduce missingness, and B, to help inform statistical analysis assumptions regarding the missing data mechanism. I hope that's helpful.
Great, thank you. I don't know if anyone has anything they want to add to that, or if I think that was a pretty comprehensive response. So, all right, let me move on to the next question. Some patients may gain or lose ability to self-report over the course of the trial, and this gain or loss of insight might be informative in and of itself. How should this be collected, and how should this data be used?
So I'll take a stab at this one, and maybe Dr. Gerard, you could follow up on my comments here. You know, it's a really sensitive question that's being asked here, and I would suggest that, you know, capturing what's important to patients while patients can self-report can happen prior to beginning a clinical trial. And so there are different stages in medical product development where we can incorporate the patient voice. And so I think that's worth considering here. In the context of a trial, you know, I think Dr. O'Neill spoke to this as well. You know, skill regression or skill decline or functional decline is really important to collect and capture. And that can be done in a number of ways. Dr. Gerard, is there anything you wanted to add to that?
The only thing I think I would add is that, you know, if it's expected that patients may lose the ability to self-report during the duration of the trial, I think that piece of consideration should be planned ahead and included as part of your standardized data collection, and then appropriately incorporated into your analysis. And that's all I wanted to mention. Thank you.
Great, thank you. I don't know if there's anyone else that has any other thoughts.
I'd just like to add one thing, Robin, to this answer, is that this is actually really important to capture, because it could also be indicative of a safety event that may be occurring if someone is losing skill and no longer able to self-report. So I know that we often focus on efficacy here when we talk about our clinical outcome assessments, but from a reviewed perspective, this is a very informative information to make sure there's not something else that's occurring because of the treatment. And so it would be extremely important to capture, particularly if it was not an expected, for example, a regression of a skill. You know, we know in a lot of our rare diseases, we do not have complete natural history, so we may not really understand the complete timelines of if it is a disease or disorder where regression of skills will appear. But this is definitely informative information and can be used just beyond the sense of efficacy, but could potentially inform safety as well.
Great, thank you. So our next question is there a way to measure saturation in social media? And I don't know, I know Dr. Daniels, Dr. Wilgatz, you both kind of spoke a little bit about social media. I don't know if you have thoughts on if there's a way to measure saturation.
Yeah, I can start anyway. So yes, given that your focus may be on qualitative data, then absolutely you can apply the same saturation methods to social media data. I guess the question would be, we typically use saturation because we're often limited by small samples, and we do it in an iterative fashion. So I think if you're running a social media analysis in an iterative way, and perhaps it's hard to reach populations or limited data online, then saturation analysis would make sense. At scale, you know, on a chronic common indication, it perhaps doesn't have the same amount of value. But yeah, the short answer is yes, you can do it in a very similar way, but it may not be appropriate depending on your method.
Yeah, and I would agree with Tom as well, in terms of if they're doing it as sort of like a qualitative approach, if they're looking at, you know, comments or social media listing in terms of text. I mean, there's ways to code data as you would code, you know, patient interviews in terms of, you know, collecting certain themes and seeing if that theme is sort of saturated over amount of comments or, you know, text that you're reviewing. So I agree. There are some approaches to do so.
Great, thank you. All right, let me turn now to Dr. O'Neill. A question came in. Can you share a little bit about the impact the preference study had on regulatory decision making?
Sure, thanks for the question. I have to say, while I don't know specifically how the agency might be using the data from our study in the context of, you know, private sponsor interactions and considerations, I do know that the agency was very positive in its feedback about the study, and when we presented the results back to the agency. So, I trust that they're hearing the patient voice, come through in that study and using it where they can in future decision making. I'll also say that we have seen a big impact from the study and the publications that have come from it so far in future trial design. So trials that have begun to be designed, since this work was done, it's been really important in informing those.
Great, thank you. Sorry, I keep muting myself and then having to re-find the mute button. Apologies for the delays. Let me turn to Dr. Gerard. Do you have any recommendations for getting this feedback when the content validity is promising but not final? We have had experiences where novel therapeutics are being considered, but FDA did not comment without content validity research being finalized and agreed to. I don't know if you have any thoughts on that, or
Yes, sorry, I had to find my mute button too. So I think I'm going to try to answer this question in conjunction with an earlier question directed at me about what type of questions would I prefer to answer. So my answer to that is also a very hard question. I'm not sure I have a direct answer for you. And as I stated earlier, in my other my other answer is that me presenting these examples, I'm not trying to say, discourage people from asking these type of questions. We're used to answering hard questions. That's what we do, right? So I think, so the important thing is we really should consider feedback from the review divisions, and each situation is unique, as your experience would tell you. And like many parts of the clinical research outside the COA space, the unknown can present planning challenges. So we try our best. When I get these type of questions, we try our best to answer, that we can provide useful comments for you. And if that requires us internally discussing with our other colleagues regarding the establishing the content validity and other issues related to those, we try to follow up with additional advice letters and things like that. So we try our best to help you to make sure that your program can be successful. Thank you.
great, thank you. So I know we're coming up on kind of the last few minutes. Maybe we'll throw out one or two more questions. Let me start with this one. Regarding use of social media data to understand patient experience, what is meant by automated searching of social media data? Could this be described a bit further, including any successful use cases? That is a big question. I don't know if anybody has thoughts on that.
I'm happy to start, Robin.
Great, thank you.
Yeah, and I have to say I am not an expert in end-to-end social media data, so forgive me. But I guess there's two approaches that people have taken. There's the quite purposeful social media listening exercise that often uses a protocol and is a lot more manual and hands-on when it comes to generating data and analyzing that data. But we've seen from many other industries, market research is a great example, where you can set up automatic listening tools that are able to essentially mine social media data in real time and give you insights in a way that doesn't involve somebody being so hands-on. So that's what I mean by automated, that it can be very passive in terms of generating data. I don't have any specific case studies I can share, but yeah, I think we're moving from a space of quite manual social media listening to a space of more automated. But perhaps Dr. Daniels has some thoughts as well on this.
Yeah, I mean, I don't think I have anything else to add in terms of automated search. I think Tom probably has more experience with that than I do. So I guess I'll just leave it at that.
Great, thank you. And I'm going to round out the the question and answer kind of panel with the following question. For those of you who spoke on specific diseases and illnesses, especially for case study purposes, could you please speak to how prepared you were or not to champion this specific disease or illness moving forward beyond the case study purposes? And I don't know, Robin, if you wanna start with that, or?
Sure, I'd be happy to. So I certainly spoke to that one case study in irritable bowel syndrome with constipation. I think that did position myself, as the rest of the project team, in a good position to have a great understanding of the patient experience. Internally, I think it's this type of research that allows us to be those kind of patient advocates on the inside, right? So when we're sitting on cross-functional teams, we can bring to life their experience and look for opportunities to embed it in drug development. I think externally, we've certainly been able to advocate for, the qualification of the measures through the CPAP Consortium and the importance of these as gold standards to hopefully really standardize measurement in this space and allow for better comparisons across treatment options, which will only hopefully beget better decision-making by clinicians and patients. But certainly I did mention it was a cross-functional effort, and we did have the IFFGD organization, a patient advocacy group in functional diseases as part of that. And I think ultimately they really have been those true advocates, right, externally, and continuing to raise the awareness of the patient experience. So I hope that, beyond our purposes of advocacy, that it does also serve the broader population, and that groups like IFFGD have found, usefulness of it in their constituents. But interested in others' thoughts and perhaps how they've leveraged similar case studies to advocate.
I'll speak to that a little bit. In the case of Sanfilippo syndrome, I think I mentioned that it's really been an important tool in informing future clinical trial design, but even it has been important in us identifying where there are gaps to where we don't have existing measures or not fit for purpose. And we've started to then be able to develop new measures around video outcomes or around stool symptoms or things that really we didn't have tools to use before. So it's been very helpful in guiding that process as well.
Great, thanks so much. Before I turn back to Shannon, does anyone else have anything they want to add? Okay, hearing nothing, I just want to give a great shout out to all of our amazing panelists who answered some challenging questions, and I really, really appreciate it. Let me turn now back to Shannon, and we'll kind of hear for our plea regarding the docket and just a few closing comments. Thanks.
Yes, thank you all for submitting your questions, and thank you to our panelists for their contributions to the Q&A session. This is another reminder that this series of meetings has an associated public docket, and the docket is open to the public and will close on September 23rd, 2022. To submit a comment, please access the docket using the link on this slide and click comment. I would like to thank everyone involved in the planning of this meeting, and I would also like to thank our presenters for their participation. Have a great afternoon.